extracellular vesicle, you can use them as sources of biomarkers because you can isolate them from the blood and other body fluids. So you can actually sort of look at the content of these and compare them between healthy and diseased individuals. Yeah, so these vesicles are known to cross the blood-brain barrier in both directions. So that opens up another possibility of using them to deliver therapies into the brain, for instance. We were talking about sustainability and, going, geez, we use a lot of plastic in the lab, there must be something we can do.
You're listening to the Neurochemistry Podcast, presented to you by the International Society for Neurochemistry. Thanks for tuning in. Let's get started.
Hello, everyone. Welcome to the Neurochemistry Podcast, where neurochemistry is for everyone. I'm your host, Nawon Kim, and today we have a special guest, Dr. Andrew Hill. He is from Victoria University in Melbourne, Australia, and he's here to talk about his research and life as a scientist. In brief, Dr. Hill did his PhD in Imperial College London and postdoctoral fellowship in UCL and University of Melbourne before setting up his lab in Victoria University. His lab has pioneered the study of extracellular vesicles, especially leveraging extracellular vesicles as markers for neurodegenerative disorders and their therapeutic tools. Welcome, Andy.
Thank you. Thanks for having me.
How are you?
I'm good, thank you. It's been a great meeting so far.
Would you like to tell us about your research?
Sure. So I guess I spent my whole career studying diseases of the brain. So my PhD was looking at a group of neurodegenerative diseases called prion diseases. And at the time, they were in the newspapers a lot because I was in the UK and working on a disease called bovine spongiform encephalopathy, or mad cow disease. And I got really interested in how proteins misfold in the brain and cause these diseases, and also looking at ways to try and diagnose them in living people. And that theme has sort of kept going throughout my whole career. And so I moved to Australia in the year 2000 and set up my lab, where I expanded to start looking at other neurodegenerative diseases such as Alzheimer's and Parkinson's disease that have very similar properties. And then about 2004, we started looking at small particles called extracellular vesicles and looking at the role that they can play in spreading these misfolded proteins in the brain. And it was actually at the ISN meeting in 2005. I think that was the first time I presented some of our work on EV. So I've got a nice connection with ISN. I've been to several of these meetings over the years, and at that meeting I got one of the Young Investigator Awards. So I found my slide deck from that presentation recently, and sort of the last slide was around our work on EVs, and that's really been my main focus for the last 20 years.
Right. So did you say misfolded proteins are in the extracellular vesicles? Or
That's right. So we can find a lot of the proteins associated with these brain diseases are released by cells attached to these extracellular vesicles. They can travel from one cell to another. And in the case of prion disease, that's how the disease can spread from cell to cell by the cell releasing the prion on one of these vesicles that gets taken up by a neighbouring cell. And Alzheimer's and Parkinson's diseases and ALS as well as another one we look at, all have similar properties.
Right. I find it interesting that it's released in the form of vesicles, so it's packaged when it's released. But there are different ways for a cell to degrade proteins in themselves using lysosome, or they could have released the proteins just by themselves, not without any packaging. So how does this packaging work?
So that's a great question. And, cells remove unwanted proteins in a variety of different mechanisms, and releasing them in these vesicles is one way. And in fact, that was what people thought the main function of these vesicles was originally. It was the way of a cell getting rid of proteins it no longer wanted. But more recently, over the last sort of 10 to 15 years, Other functions of these vesicles have been described. So it's a way of intercellular communication, because when you look inside these vesicles, there's proteins, lipids, metabolites, but also genetic material in the form of small RNAs, which has become a big focus for our lab as well, and also DNA. So these vesicles have multiple functions. One is to get rid of things the cell doesn't want anymore. Another is to send out messages that are picked up by other cells as well. And so, they have multiple functions, and the cell can decide, depending on the kind of vesicle. There's lots of different flavors of extracellular vesicle. They're formed in a couple of different ways. But the cell can actually decide what it wants to package in these little bubbles that are then sent out by the cell. So it's quite a complex process.
Right. So it sounds like it's a very versatile structure. It does a lot of things. It could be used as a way to get rid of unwanted misfolded proteins, or it could be used as messenger. But how does the cell know like what to package, and how is the selectivity achieved? Is it packed first with is it selected within a cell first and then budded outside? How does it start?
So there's one type of extracellular vesicle called exosomes, and they're formed through an endocytic mechanism. So you find in these exosomes proteins that are found in the endocytosis pathway, as well as proteins on the plasma membrane. So these are enriched. These exosomes are enriched in transmembrane proteins, GPI anchored proteins. The prion protein is one such protein, and also as the process, so you get the sort of inward budding of the plasma membrane. That's the first part of this process. It also captures a bit of the cytosol of the cell at the same time. So you're getting a bit of an insight into what the cell is doing at a particular time. And so applications of looking at these EVs can actually help diagnose diseases, because these particles are being released by cells, and they're giving you a bit of a snapshot of what's happening in the cell at the time. So you can use them, and this is work that we've done, as sources of biomarkers, because you can isolate them from the blood and other body fluids. So you can actually sort of look at the content of these and compare them between healthy and diseased individuals.
Right. Did you say blood? Does it cross blood-brain barrier?
Yeah, so these vesicles are known to cross the blood-brain barrier in both directions. So that opens up another possibility of using them to deliver therapies into the brain, for instance. So you can engineer vesicles to have properties on the outside that sort of enhance their ability to cross the blood-brain barrier and deliver their cargo into the brain. And we use EVs that we isolate from the blood to diagnose Alzheimer's, Parkinson's, and other neurodegenerative diseases by looking at, we look at small RNAs. We look particularly at the expression levels of microRNAs in the vesicles we can isolate from the blood, and we can get really nice diagnostic readouts by doing this. So not all of those EVs we're picking up in the blood obviously come from the brain, but there's enough of a signal there that can help us diagnose these disorders.
Right. It sounds like a really powerful tool, especially less invasive as well compared to other possible treatments. So we were talking about how it's generated from cells, but what about the receiving cell? How do they receive it? Does extracellular vesicles stay in the extracellular area, or does it dock to another cell, and is it internalized to a different cell?
Yeah, so the vesicles can, they can dock with cells. You can get membrane fusion, and that will pull apart the vesicle, and the contents will be delivered to that cell. There are some receptors that can be involved in this transfer as well. But yeah, there's multiple methods by which these vesicles can bind to and be taken up by recipient cells. And when that happens, their cargo is delivered, and it can have some sort of functional activity as well, whether it's through the proteins or the microRNAs or small RNAs that are delivered to the recipient cell.
Right. And how do you study extracellular vesicles in your lab? Do you do it in culture? Do you do it in slices? How do you catch them?
Yeah, we do sort of all of the above. So we do. So I'm a biologist by training, but also I'm interested in how we can use our research to translate that into helping people with these diseases. So we do a lot of cell culture studies, and so you can. And these particles are tiny; they're about 100 nanometers in diameter. So you can't see them with traditional microscopy. You need to use electron microscopy or some specialized biophysical instruments. So we use cell culture studies. We've also done some work with brain slices, isolating EVs released from brain slices. We also developed a technique for isolating EVs from brain tissue, so from frozen and fresh mouse or human brain tissue. And then also we isolate EVs from biofluids such as blood, CSF, urine, saliva, and because of their nature, they're actually really quite they're quite hardy. So you can take samples, frozen samples that have been in your freezer for a long time, and you can still isolate EVs and do experiments with them. So yeah, we've pulled them out of lots of different things.
Nice. And how common are they? Are they abundant in tissues, or do you have to induce to study them?
So being very tiny particles, you need to sort of get enough material to be able to use in your downstream, experiments. So, we can isolate them from, 0.5 to 1 gram of brain tissue. We can isolate enough to do some genetic analysis from, 0.5 to 1 mil of blood. Cell culture, you need to grow quite a few cells. So it's not uncommon to see people in the lab have whole shelves full of culture flasks growing enough cells to isolate enough EVs for their experiments. So it depends on the system. Some biofluids, CSF for example, don't have that many EVs in them. So obviously you need to use a larger volume to do those kind of experiments. Yeah.
And we were talking about the quantity, and I also want to think about the distance they can travel. So you said it's in blood, it's also in urine samples as well. So does it have a specific destination, or do you think it's just a drainage from the cells?
I think it's probably drainage from the cells. I mean, there might be some targeting of specific EVs. They might have a particular component on the outside that targets it to a particular cell. But within our blood, there's billions of these particles circulating at one time. And so I think naturally there's just going to be a flow of these vesicles around the body. And maybe it's some sort of random event that it finds a cell that it binds to and releases. I guess in tissues, the release is going to be contained within that particular tissue. So I think you'll probably get the uptake more there. But yeah, you've got them circulating throughout all of your biofluids. So I guess they're just circulating until they find a home.
Right. So it sounds like a really ubiquitous, like it's omnipresent. You can find it anywhere in the body, and it can be used for diagnosis of neurodegenerative disorders. So do you look for different markers for different diseases, or do they have a common a common marker that could be used to tell if a person has neurodegenerative disorders or not in the EV?
So yeah, lots of labs are looking at using EVs as a source of biomarkers for diagnosis in cancer, infectious disease. We and others are doing it in neurodegenerative diseases. And in our case, we look at microRNA profiles. And so basically we have panels of microRNAs that have different expression levels that can diagnose a certain disease. And so what we're trying to build up is like a dementia panel. And while we found a few microRNAs that seem to indicate that there's some neurodegeneration happening, there are quite specific ones that tell you what kind of disease you have. So yeah, it's a combination of some specific ones and then some ones that seem to be common across all of these neurodegenerative diseases, which you'd probably expect given they have, similar pathways.
Right. Okay. Moving away from science, I would like to talk about going green, because I noticed your group was the first lab in Australia to achieve a green lab certification. What was this certification, and how are you making your lab more environmental friendly?
So, like I mentioned before, we use a lot of cell culture plasticware. Most labs use a lot of plasticware. And this sort of came about during COVID, and I had a fantastic postdoc in the lab, Edward Willems, and, we were talking about sustainability and, going, geez, we use a lot of plastic in the lab, there must be something we can do. And we came across this organisation called My Green Lab that runs a number of programs to help labs develop more sustainable practices. And they also work with various vendors as well to sort of, look at recycling programs. And so the process was we got the lab involved. We had a few lab meetings. We talked about it. We all acknowledged, how we could, be a bit more sustainable. And to get the certification, we had to do a survey where every member of the lab was surveyed for their awareness of things like electricity usage, water usage, and even travel to conferences and things. It covered so many different aspects. And then we got a score back and we went, there's a lot more we could do here. And so we went through a program of changing some things in the lab, and it included things like we realised our autoclave which was a departmental autoclave, was left on all the time. And there was no one could answer, why do we leave that on all the time? And then when we talked it through, we found out that it only took 10 to 15 minutes for the autoclave to actually warm up. So we changed the practice to switching it off when it wasn't being used and to switching it on 15 minutes before you needed to use it. And there was an instant power saving. Even little things like the taps, putting those, I can't remember what they're called, but you can put these flow restrictors on the taps so you can restrict how much water is being used. Even discussions about minus 80 freezers. When I started my PhD, they used to be called minus 70 freezers because they're at minus 70 degrees. Now they seem to be at minus 80. And again, no one could figure out why have we gone from minus 70 to minus 80. So we decided to switch some of our freezers back to minus 70. It doesn't really change anything about the storage, but again, it's saving power. And, if more people take this on, it actually creates, really big sustainable savings. And then there were the sort of other things around recycling plastics where we could. Obviously, things that are biohazardous you can't recycle, but like the little wrappers you get on your plastic pipettes, we put special bins in the lab where those could be recycled. So it was lots and lots of little things. And then after about 6 months, we went and redid our survey, and we'd shown improvement across all the relevant areas. So we got certified as level green, which is their highest level. So we're really proud of that because it was a great thing to do, and Edward really pushed the lab along and understanding how important this was. And so every 2 years we have to re-go the certification, and new members of the lab come and join and learn about these practices, and it's sort of spread across different labs. And now at the new university that I'm at, Victoria University, we've got our first green lab certified there as well. So we're spreading this around.
Yeah, that's really inspiring, because I think there's not enough discussion about going green in the labs. Research is not very environmental friendly. Like, we go through tips and 15 mil Falcon tubes so much, and I've always felt guilty just tossing them into the bin. And as you said, disposing them in a more environmental friendly way could be really helpful. I think not many people are very cautious about how they dispose of stuff, like what can be biohazard and what is less of a hazard and thus can be recycled. And some people would be just blasé about everything and throw all the liquid waste down the sink. Yeah, I think we need more discussions in the lab and across different labs how to make our labs more environmental friendly. And like the energy aspect as well, because these big machines like freezers and autoclaves, because they usually are taken care of by the department, not individual labs. We just leave it to them and assume they're taking good care of it, and yeah, we just leave it to other people. But I think we should be more proactive as in like what we turn on and when should it get turned off. And traveling as well. People go to conferences and people usually prefer flights, but like taking trains as much as possible and using public transport if possible to the lab is also very important, I think. Yeah, because that's one thing. I just moved to the US and I noticed a lot of people drive and car is almost a necessity. But I would love to be in a society where I can comfortably take tram, buses into the lab and not feel so less bad about the environment.
Yeah, it's all the little things I think that count. So, and I think that's what we found through this process. It's things that you probably wouldn't think of. And it's funny, I ended up taking some of these things home and sort of going, oh, we should be turning this off at home when we're not using it. So we've done a few things at home as well to improve our sustainability.
Yeah, it seeps into your life. Like once you start making small changes and you notice things that you didn't think about before. Yeah, that's really nice.
And look, I think it's a great scheme. So, I urge people to sort of take a look at the program. And, we found that our institution got right behind it. They even helped change the light bulbs in the lab to LED lighting. They were going to do it anyway, but this is one of those things that was going to be a quick win for us. So they helped with that. And yeah, it was amazing. Everyone seemed to just jump on board and now it just becomes sort of second nature, a lot of the things that we do. So, yeah, it was a great process.
Do you know if this certification is Australia only or is this an international?
Oh, no, it's a global.
Right.
So My Green Lab, I think, is a US-based organisation. So they've got a great website. They've built a community. So, you can share ideas and they engage a lot with vendors as well to help with, returning those eskies that you get your cold shipments and things like that. So I think it's great just to see the engagement from everyone, from our students through to the companies that we deal with. So yeah, it's great to see that happening.
Amazing. Yeah, I will definitely bring it up in my lab meeting. Okay, I think it's time to move on to my last question. What makes one a good scientist? Because as we are summarized as one word scientists, but we are required to do a lot of things, just designing research, executing research, reporting research, and also teaching other fellow colleagues and communicating to the public and to our other science communities as well, and so on. What do you think is the most important quality to be a good scientist?
Oh, that's a great question. Because like you said, there's lots of different aspects to being a scientist. I think if you ask someone on the street, they just think you're just in a lab playing with test tubes, not doing all those other things that you've just mentioned. I think for me, it's about being curious. I think, I think it's a privilege to be a scientist. And I think what got me going was just that buzz of knowing I'm going in the lab, I'm finding out something for the first time that perhaps no one else has seen before. So having that curiosity-driven approach, I think, is really important as a scientist. And it's also a kind of a strange career in a way because we get a lot of training around how to do experiments and things, but there's a lot of things that you kind of just sort of learn along the way. So I think resilience is another key because, as we all know, sometimes experiments don't work out, our publications get rejected, we don't get our grants, and those moments are really, really tough, but I think there's always learnings from that as well. So I think it's about being curious, open to learning new things, staying the course, and I think making good networks around you, not only for sort of support through those tough times, but also you learn a lot from other people as well. So that's why coming to conferences, presenting your work, talking with others is a great way of really engaging more fully with science. So that's what I think it's about.
I'm going through exactly what you said, because I am starting my postdoctoral training right now, and I'm learning that I experience failures a lot more than successes. So I will need resilience and my support group and curiosity to stay passionate despite all those failures.
Yeah, and it can be tough at times, but I think generally it's a great career, and like I said, you learn a lot through those tough periods. I think you learn a lot about how to navigate different problems, and it's that whole curious thing as well. If you're doing stuff that's been done before, that's easy, but it's not really going to help you as a scientist. I think designing good experiments and chasing big problems, I think it's not easy, but it's so rewarding when you get there.
Yeah. Thank you so much for the empowering and inspiring talk. Thank you very much.
Thank you very much.
Thank you for listening. Don't forget to follow and subscribe for more neurochemistry talks. Till next time.